Welcome back. Today we are tackling plant transport. That means everything from the roots to the xylem to the phloem. We're going to go way more in depth than we did last time. And we're also going to touch a little bit on some old experiments that are super unethical nowadays. So to start off, we're going to talk about roots. So if you remember from last time, roots' main purpose is anchorage and water absorption. That's the biggest role that they have in a plant's life. Some roots will branch into smaller roots. They could go far in the soil or not super far. The branched roots can also grow root hairs, and all of this is just to maximize surface area. The more surface area a root has, the more absorption they can do, meaning the plant is happy. In the picture to the left, you see a dicot taproot. What that means is that that root has a very thick root, singular root, that goes really deep into the soil. A taproot is this idea of a singular root, whereas a fibrous root is a root that doesn't have a large middle root, but rather just a bunch of equally sized roots. So a dicot taproot is used by dicots, obviously, but also the types of plants that need really secure anchorage. So think those really big trees outside, they need that dicot root. They reach deeper, they really anchor the tree, and they absorb a ton of water and mineral ions. There are 4 ways that minerals, ions, water can travel into the roots. The first way is diffusion. Now, if you don't remember osmosis and diffusion, you should pause this video and maybe go review your notes from last year. I'm going to do a super quick overview of it, but I'm going to be talking about it a lot. So if this is something that you didn't get last year, I really suggest you take a minute and just refresh your memory on what diffusion and osmosis and active transport is. For a quick overview, osmosis is the movement of water from a high concentration to a low concentration. The idea of osmosis is to equalize the concentration of water on 2 sides of a concentration gradient. Diffusion is the same exact thing, except instead of water, we're talking about literally anything else. Minerals, nutrients, sugars, anything that is not just water is classified under diffusion. Again, this is a passive transport, no energy, moves from high to low. Active transport is kind of the opposite. It uses energy, and it's when you're trying to move minerals, ions, whatever you want to be moving from low to high, meaning that there's already a high concentration of what you're moving in the area that you're looking at, but you want more. So building on an already high concentration with the use of energy. Okay, that was my quick recap. If you still are a little confused, again, pause this because I'm just going to get further into all of this. So let's get back to how we bring things into the roots. Number 1, diffusion of mineral ions. This is slow. It's from high to low. It's passive, no energy. It just wants to slowly bring all those minerals into the roots and eventually to the stem and to the leaves. Our second option is using fungal hyphae. So fungus and trees, plants, whatever, have this mutualistic relationship, meaning that they both benefit from this relationship. Because a fungus can grow wide and far, it kind of teams up with the plant roots. So the fungus will grow further than the roots, which allows for a wider and more comprehensive area that's connected to the plant. And the fungus will uptake all of these minerals and pass it along to the plant, so the plant doesn't have to do quite as much work. In exchange, the plant gives the fungus sugars, extra sugars, so the fungus doesn't have to do as much work to get fed. It's mutualistic. It's been around for a very long time, and it seems to work really well for both involved. A third way is mass flow. So mass flow is basically just a lot of things traveling in at once. Minerals can kind of hitch a ride with water as water is being pulled into the roots. Hydrostatic pressure, which is water pressure, is generated when the roots continuously suck up the water. And then active transport. So again, if a plant really needs a high amount of a certain mineral but already has a lot of that mineral, it's going to use energy to force that mineral into the plant from a low to high concentration. So opposite of diffusion, right? Diffusion is no energy, active transport has energy. You have to use this to get more than you need. Moving into the xylem. So all of what we've talked about so far is in the roots, but now we're in the xylem. This is a theory that you should know by name, cohesion tension theory. This is how water moves throughout the entire plant. You've actually probably seen something like this and you didn't even know it. So cohesion is a water property, meaning that water sticks to water. Have you ever been driving in the car and it's raining outside and you're looking at the window and these raindrops are racing down the window and you picked one that's hoping to win the race, and as it moves it connects with other raindrops that are stuck on the window? Yeah, that's cohesion. Water properties, it's everywhere. So the idea in plants is that water will always be pulled out of the leaves through transpiration. And I know we've talked about this, but transpiration is the loss of water. So when a leaf will get hot enough, like in the sun, the water that's in the leaf will start to turn into water vapor. Water vapor will leave the leaf and go into the atmosphere. Again, this is osmosis at work. There's a higher concentration of water vapor in the leaf, so it wants to move to where there's a lower concentration outside to hit equilibrium. When the water vapor from in the leaf moves to the atmosphere, that water needs to be replaced. So the leaf pulls the water from the xylem. As the leaf pulls the water from the xylem, now the water in the xylem needs to be replaced. So through cohesion and adhesion, again, cohesion is water sticking to water, adhesion is water sticking to anything else. In this case, water sticking to the walls of the xylem. So through that, water is continuously pulled up from the roots into the xylem and then into the leaf. This tension or pressure occurs because the loss of water in the leaves. It's just kind of a domino effect. As the water leaves the leaves, the water needs to be replaced. So then the water is replaced from the xylem. Well, now that water needs to be replaced. So then the water from the roots goes into the stem. Okay, well now that water needs to be replaced. So water is constantly being taken up by plants because water is constantly being lost by plants. So let's talk about at night. Because we know the sun doesn't shine at night, which means that water vapor isn't being formed at night. And if water vapor is not being formed at night, then why are plants still losing water? Well, it's still going to lose water just because this pull, this tension pull, is still happening. So some water is still going to be released, even if it's not 90 degrees out and the sun beating down on the plant. What happens is the root cells continue to pump minerals into the xylem, and as they pump the minerals into the xylem, because of that mismatched gradient, water is going to come in with the minerals so that there's a little bit more of an equilibrium. As that water gets pulled in by the roots, it's going to continue to move up the stem, into the leaf, pushing sap, generating pressure. It's just a never-ending process in a plant. We've talked about this before, transpiration rates. We've talked a lot about them in the last lecture. It wasn't on a slide that was pretty much dedicated to it like this, but I brought it up a number of times, so I'm not going to go through this bullet by bullet. A couple things just to remind you of what we talked about is for the biotic factors, remember when we talked about cacti, xerophytes, the thickness of waxy cuticles, right? Cacti have very thick waxy cuticles to prevent water loss. So the thicker the waxy cuticle, the less transpiration rate. Whereas in your abiotic side, the more light that shines on a plant, the higher the transpiration rate, right? We talked about blue wavelengths last time. So we've talked about most of these in the last one. You can rewatch that video or you probably have it written down. So take a peek, but this is just kind of a reminder. There are a number of factors that affect transpiration, and you should absolutely be able to pull a couple if I asked, what are some ways that transpiration is affected? So now we've covered roots, we've covered xylem, we're moving into phloem. Phloem transports pretty much everything that's not water. So sugar, amino acids, hormones, mRNA, different chemicals, and as mentioned previously, phloem transport is bidirectional. That means it can move up towards the leaves or down towards the roots. Xylem only moves one way, from roots to stem to leaves. So before we get into the fancy pressure flow hypothesis, which is how the sap moves, we're going to talk about active translocation. These are just terms that you're going to need to know to understand the next slide. So active translocation is the movement of sap. I'm going to call the phloem products sap from here on out. So that could be sugars, chemicals, minerals, anything. But I'm going to refer to it in general as sap. So active translocation is the movement of sap from one location to another using energy. So where the sap starts is called the source. So this is where all of those ingredients of sap are stored. So your carbs, your sugars, etc. They're stored in the source. They are then brought into the phloem and moved, whether up or down, and unloaded in the sink. So starts in the source, ends in the sink. The sink is kind of the end goal of wherever in the plant needs those different products that are found in the sap. Okay? So now let's jump into the pressure flow hypothesis. Remember that the xylem, that movement of water is called the cohesion tension theory. In phloem, the movement of the sap is called the pressure flow hypothesis. It will never be referred to as the movement in the phloem. It will always be referred to as pressure flow hypothesis, or if we're talking about xylem, the cohesion tension theory. You should really highlight that or underline it and make sure that you understand that cohesion tension equals xylem, pressure flow equals phloem. So, we talked about active translocation on the last slide, but number one, the first step here is talking about that again. So organic compounds, aka the sap, are pumped into sieve tubes using energy. Once that sugary sap is in the sieve tube, the water from the xylem is like, hey, wait a minute. I don't feel like we're on equal ground here. I'm going to do osmosis and diffuse my water from the xylem into the phloem to equal it out. Once that water gets into the phloem, wherever that sap buildup is, the water pressure increases. When that pressure increases, it starts to push the sap in whatever direction the sap is moving, whether up or down, until it reaches the sink, and then all that sap is unloaded from the phloem into the sink. Once the sap is gone from the phloem, the water then returns to the xylem. Now I mentioned a few times that the sucrose is not, or the sap is not pumped directly into the sieve tubes, but rather it's using energy to get itself into that area. So it does this by something called chemiosmosis. So what a shocker, the term osmosis is showing up again. This is massively important that you understand the process of osmosis. In this case, it's also using proton pumps and hydrogen ions. So what happens is hydrogen ions create a proton gradient. So if you look in the picture, hydrogen ions start on the bottom here, where there's a low concentration, and using energy, they're pushed out to where the sucrose is, and then they come back in through this co-transporter protein, and sucrose kind of just hitches a ride. It's like, hey, yeah, sure, I'll join you. And when this gradient starts to happen and the hydrogen ions get pushed through and back and through and back, the sucrose and the sap that's formed from the phloem come back as well. And that's how the sap moves from the source into the phloem. But there's always more. Sugar doesn't just flow into the phloem. It doesn't just like open a door and say, hey, I'm here, let me in. It can only go through certain cells, or can only go through certain openings in the cells, and those openings are called plasmodesmata. So, again, the sap that's in the source can only move into the phloem using chemiosmosis, which is this active transport using energy. And it moves into the phloem through plasmodesmata, and then it goes through the entire pressure flow hypothesis and moves to the sink. I know this is complicated, but if you missed a piece of this, that's the beauty of videos. Just pause, go back, and rewatch it. Now, one of the last things we're going to talk about today is aphid experiments. Aphid experiments are experiments that happened in the 40s, so a very long time ago, when ethics didn't really exist. In this case, they used radioactive carbon dioxide and made aphids eat it to determine sap flow. But before we get super into that, let's talk about what aphids are and what they did. So aphids are an insect. And the way that they survive is they kind of stick their stylet, which is, you can kind of see it in the upper right-hand corner picture. Stylet is like a really long nose, and they stick their stylet through plant tissue into phloem. And then they suck up all the sap that's in a phloem and they eat it. The way that these experiments ended up working is that they let aphids eat the plant phloem and then knocked them out and cut their stylets off. And then looked at what was in the phloem from their stylet and then analyzed that. To get a little bit more in depth with this experiment, we're going to talk about what actually happened. So again, like I said, in the 1940s, you had radioactive carbon dioxide, and you gave that to the plants. And then the plants used that in their photosynthesis process. By using that radioactive CO2, all of the sap that was created was then radioactive. What the scientists wanted to research was how that radioactive sap moved throughout the plant. So at different time points, they let the aphids eat the plant, and then they knocked them out, took their stylets, analyzed the phloem to see how quickly the rate of sap was moving. And they could tell because some of it would be radioactive and some of it would not be. And so they kept doing this until the radioactive sap was everywhere in the plant. And that's how we figured out how phloem moves sap, which is through this experiment. Of course, nowadays you would never do this because it is not ethical to kill aphids just to do your experiment, and you could easily do this with a syringe, but here we are. This is how we learned. The very last thing that I want to cover is kind of just a quick overview. Make sure that you really understood what we were talking about. So, for plant transport of water, roots absorb water, water is transported up into the xylem, and then transpiration creates a force that pulls that xylem sap, but we know that in xylem it's water. So transpiration creates a force that pulls that water up and out. So water starts in the roots, goes to the xylem, is pulled out through transpiration. For different gases, leaves exchange CO2 and O2, right? Carbon dioxide and oxygen. And roots exchange gases with any sort of airspace they might find in the soil. And then sugars are produced by photosynthesis and then transported as phloem sap to the roots, to other parts of the plants, to anywhere that needs these chemicals or minerals or sugars, whatever they need, because phloem is bidirectional. It can move up or down. So that is it for today. Thanks for tuning in. I'll see you next time.